Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about tyrosine kinase receptors. So in our previous video we talked about G-protein coupled receptors and how that is a very common signal transduction mechanism used in a variety of different signaling pathways. For the vast majority of processes in physiology that we're going to be talking about in this course, we're usually going to be talking about G-protein coupled receptor pathways, but the purpose of this video is to introduce you to another fairly common signal transduction pathway, which is the tyrosine kinase receptor, or RTK pathway. Not going to be quite as common as the G-protein coupled receptor pathway, but you will see this from time to time. So here we can actually look at some common RTK pathways, and I have highlighted here the insulin and insulin receptor pathway because that is one coming up here in a few videos when we start talking about the endocrine system and specific hormones. This is going to be one that we see quite a lot of. So in this particular case, insulin is the ligand, the hormone, that activates the insulin receptor, and notably the insulin receptor is not a G-protein coupled receptor like we saw in the previous video. It is a tyrosine kinase receptor, which we will introduce in general terms here in just a minute. So, as you can see here, when insulin binds to the insulin receptor, the overall effect is that glucose is allowed to be transported into cells, and glucose can be stored up or used right away. The specifics of how that works in particular is something that we'll cover in a later video, so let's not worry too much about it for right now. The other RTK pathways that you can see here, the thing that you'll see they all have in common with each other is epidermal growth factor, vascular endothelial growth factor, insulin-like growth factor. So you see the term growth factor is a common denominator with these different types of RTK pathways. So other than the insulin receptor pathway, receptor tyrosine kinase pathways tend to be those that induce cell division, cell growth, cell proliferation, things of that nature. Now, these are all a little bit different from each other in terms of what the overall desired effect is, whether it is just growth and differentiation of stem cells, creation of new blood vessels or lymphatic vessels, body growth, whether it's the lengthening of bones. These are things that we may talk about here and there throughout the course, but the thing that they all have in common with each other, obviously, is that they are there for some type of cell division or cell growth pathway. So if in the future we happen to be talking about cell division or cell growth, you can be fairly confident that an RTK pathway is likely behind how it all got started. Okay, so what we're going to do in this video is just cover the bare bones basics of how a tyrosine kinase receptor pathway works. So tyrosine kinase receptors generally consist of two different subunits, each with a ligand binding domain and then an intracellular kinase domain. So let's go ahead and label these. You can see the ligand binding domains face towards the extracellular fluid, which is going to be where the growth factor or hormone or ligand is going to come from. And then the kinase domains face towards the intracellular fluid, where all the mechanics of the inner workings of the cell are located. If we're going to induce some sort of response like cell division, we need activation or deactivation of proteins inside the cell. Okay? So we have our ligand binding domains here in the extracellular fluid, and then facing towards the intracellular fluid are kinase domains. You'll recall that a kinase is an enzyme that transfers a phosphate to a protein, but for right now, these kinase domains are inactive, and they will be inactive as long as there is no ligand. So in the absence of ligand, these two subunits stay separate from one another, and those kinase domains remain inactive. But let's take a look at what happens when the ligand becomes available. When the ligand binds to these receptors, we're going to see dimerization of these two subunits, in which that means that the two separate subunits come together and come in close proximity to one another. And what this is also going to accomplish is it will activate the kinase activity of each subunit. Now, what's going to be interesting about what happens here is we see kind of a unique process happening. This is called transauthophosphorylation. If that sounds like a mouthful, that's because it is. But what trans-autophosphorylation is that the kinase domain of one subunit starts transferring phosphates to the other subunit, and then the other subunit transfers phosphates to the first subunit. So basically, these two kinase domains phosphorylate each other. So why this is important? Well, there's a couple of reasons. First, activation of these kinase domains, in addition to the trans-autophosphorylation, you'll also get some phosphorylation of substrates that are inside the cell, very in line with what we've seen from the activity of kinases before. But in particular, for tyrosine kinase receptors, a very common thing we see is that these phosphorylation sites create what we call docking sites for intracellular effectors. So what I've illustrated here is a generic docking protein that, now that these kinase domains have attached phosphates, this docking protein has something to grab onto. So this is very often going to lead to what is sometimes kind of a complex series of events inside the cell that generally lead to kind of a mass phosphorylation of proteins inside the cell. So the general idea of what I'm trying to convey in this picture is that the docking protein attaches to the tyrosine kinase receptor, and this creates an activation point for a different kinase, which we will call kinase number 1. Kinase number 1 will phosphorylate another enzyme, which we will call kinase number 2. Kinase number 2, now that it is activated, will then phosphorylate kinase number 3, and then this cascade of events will go on and on and on until we eventually phosphorylate all the target proteins inside the cell that we want to phosphorylate to accomplish whatever it is we want to accomplish, whether that is cell growth, cell division, cell differentiation, whatever it may be. Now, one thing you may be wondering here is, well, why do we need all these different kinases? Why couldn't it just be sufficient to activate this first kinase and then let it just go do its business? Well, the issue is that since we're talking about pathways that are involved in cell division and cell growth, there generally needs to be a lot of guardrails. There needs to be a lot of regulation points, because one of the things you learned in general biology is that when cell division and cell growth becomes unregulated, you end up with things like cancer, okay? So what we're going to explore here, just to kind of see a specific example, is we're going to look at something called the RAS-MAP kinase pathway. This is a very common receptor tyrosine kinase pathway that usually will cause a target cell to grow and proliferate. To the point that I was making before, if we mess up the regulation of how this pathway is supposed to function, we can end up with something like cancer. So a lot of the proteins in this pathway are proto-oncogenes that are actually very commonly mutated in a lot of cancers. So in pathways like this, we generally need to have kind of these multiple kinases, these multiple layers of regulation, to make sure that if one of them does not do their job properly, if the others do their job properly, we should be okay. Okay, so let's look at the RAS-MAP kinase pathway. So this receptor here could be basically any of the receptors that we were looking at before, the epidermal growth factor receptor, the VEGF receptor. So you can see what we talked about before, that once the ligand binds, so this ligand would be one of those growth factors, whether it's VEGF or EDGF or one of those, we get dimerization and trans-autophosphorylation of the two receptor subunits. As we said, this is going to create a docking site for a protein, and in the case of this particular pathway, this docking protein is called GRB2, or GRAB2 if you want to call it that. So this is going to cause another protein to grab on, no pun intended. This protein is called SOS, which stands for son of sevenless. So SOS is a type of protein called a guanidine exchange factor, or a GEF, or GEF. So what this protein is going to do is activate a small G protein called RAS. So RAS, when it is bound to GDP, much like we saw with G protein coupled receptor pathways, is inactive. SOS, as a guanidine exchange factor, allows RAS to get rid of its GDP in favor of GTP. This activates RAS, and RAS can now lead to the activation of RAF, which is a kinase, which leads to the activation of MEK, which is a kinase, which leads to the activation of MAP kinase, which is a kinase. So you can see we have kind of a lot of redundancy here in terms of lots and lots of kinases to make sure that we have lots of guardrails to keep this pathway under control. So then MAP kinase is going to hit a lot of intracellular effectors, phosphorylate them to activate them, phosphorylate them to deactivate them, and the overall effect here is that this particular cell will be induced to enter the cell cycle, start growing and dividing to accomplish some sort of necessary task. Now, one thing I will point out here in particular, this protein here called RAS, the RAS oncogene, as it's sometimes called, other than P53, which is oftentimes called the guardian of the genome, RAS is one of the most commonly mutated genes in lots of different cancers. You may have heard of it before. If you haven't, you may want to look into it. So in introducing the RAS MAP kinase pathway, I'm not expecting you to memorize this pathway. We're probably not going to talk about this particular pathway very much, if at all, in this course. I merely am introducing this to show you what a tyrosine kinase receptor pathway looks like and what sorts of things it can accomplish when it becomes activated. So just to kind of summarize some of the roles in this pathway, GRAB2 was the docking protein, SOS was the guanidine exchange factor that activated the RAS oncogene. RAS, as I said, is a small GTPase that activates the kinase that leads to RAF, MEK, MAP kinase. So those are all necessary steps in order to get the pathway to reach its conclusion. Okay, that's going to do it for this video. If you have any, this is a list of vocabulary terms that you probably ought to be aware of after watching this video. For checking your understanding, number 1, what is the most common outcome of activating a tyrosine kinase receptor in general? Number 2, how are tyrosine kinase receptors different from G-protein coupled receptors? And you might also think about how they are similar in some ways. And finally, why would we say that tyrosine kinase receptor pathways are common targets for cancer-causing mutations? So that's going to do it for the video. If you have any questions, feel free to drop them in the comments section. Otherwise, I will see you for the next video.